Hello, my name is Shelby Harris, and I will be doing my immunology video on antibody structure. To start off with an overview of antibodies are called immunoglobulins, and there are 5 different isotopes of antibodies. The first one is IgA, which is found in blood serum, nasal mucus, saliva, and breast milk, mostly most of your body fluids and stuff like that. And it can be passed from mom to kid through breast milk and help the baby's immune system early on. The second one is IgD. IgD is, they don't really know the exact function of IgD, but it's thought to make antibodies in B cells because it's also found in B cells. IgE. IgE, you found that in small amounts in adults, mostly in the fetus. It produces, it mostly protects against parasites, but in places where there isn't a lot of parasites, like here in the United States, it has major roles in allergies. IgG is the most abundant antibody found in the body. It's found in the blood. It's really important for the antigen antibody complex with leukocytes and macrophages. So, like, the essential part of the immune system is what IgG does. And it can pass onto the fetus through the placenta. So it can be passed on from mom to kid to help the baby's immune system start out. And the last one is IgM. And IgM is found in the blood and on B cells on the outside. That's what the initial antigen will bind to the B cell to start the adaptive immune system. It has roles in making antibodies and cell signaling pathways. Antibodies in the body are produced by mature B cells called plasma cells to combat invaders in the body. So when a B cell encounters antigen on its IgM antibodies that are bound to the membrane, it will start to mature into a plasma cell, and that plasma cell will crank out antibodies made to essentially attack that invader of the body, whether it be a bacteria or a virus or something, another antibody even. And over here we have antigens and antibodies coming together. So one thing that antibodies can do with the antigen is it can cover up the antigen and block receptors. So if it's a virus that has to bind to something to the outside of the cell to infect the cell, its receptors can be blocked by the antibody itself. And then another thing antibodies can do is that they can hook all together, and antibodies can even attach to other antibodies, and that will form like essentially like a clot, and that invader, that antigen, can now not move around the body as easily. And then down here we have models of all the antibodies. So IgG, IgE, and IgD are monomers. They're just single units of an antibody, versus IgM is a pentamer. It has 5 around in a pentagram, and IgA is a dimer. There's 2 of them. The basic structure of an antibody is that you have your heavy chain, which is mostly the constant region, which are these 2 right here. These are your heavy chains, these long ones. You have the light chain, which is the variable region. It's up here. And the heavy chain also has a little bit of a variable region, but it's mostly constant. And the same thing with the light chain is that you have this variable region right here and the small constant region. You have the Fab region. So the Fab region is this top part, and it's called Fab because when they cut it and they realized that this binds antigen, they were like, oh, that's fabulous that it does that and stuff like that. So they call it the Fab region. And the FC region is the bottom part of the antibody. And they call it the FC region because when they did cut it and they left it, it fell out of solution and formed like a pellet at the bottom of whatever they had it in, the beaker or whatever. And that's what it's called, FC. It falls out when cold. And the FC region also dictates the class of antibodies. So this part right here is what tells antibody whether it's IgA, IgD, or IgM even. So the next part we have antibody variants. So we have 3 types of antibody variants. So we have isotype. Isotype is determines different species. So both humans and rabbits have IgM antibodies, but IgM antibodies between the humans and rabbits are very different. You can't use the same ones. So the isotype variants will occur in the constant regions of the light chain right here. So allotype is differences between within a species. So say one species of rabbit, I don't know, cottontails I guess are a species, will have a certain IgM antibody in there, also in their constant regions of the light chain, versus what are those Netherland bunnies that have the really long hairs? They would have something different here. So you wouldn't be able to use antibodies between the two rabbits even though they're in the same species and stuff like that. So idiotype is within an individual's own antibodies. So idiotype is between the variable region of the light chain. So even if you're an individual, you can have IgM, but your IgM is different than your IgA. And that's where the idiotype will come in to determine that. So we have our Ig domains. The domains are basically the way the beta sheets of the antibody protein folds. So they differ between the variable and constant regions of the antibody. The constant regions have 7, while the variable regions have 9. So that's how you can tell, that's how on the molecular level you can tell which one is the variable and which one is the constant region. So here we have a constant region where it has 4 going like that, and then 3 in the back, so that's 7. But here we have a variable region that has 4 going like that, and then we have more in the back. So, and those extra 2 beta sheet strands that are in there is what forms the antibody receptor region on the variable part of the antibody. So our light chains are 2 identical amino acid chains at the top of the antibody. So at the top half is the variable region, the bottom half is the constant region. So here we have our light chains, these 2 little ones right here. We have the variable region at the top and the constant region at the bottom. These constant regions can be referred to in their amino acid sequence as either kappa or lambda. Most of your light chains are mostly kappa, while a little bit of them are lambda. Lambda is more rare in certain animals. In humans is almost half-half, but in like rabbits is significantly less than the kappa. The hypervariable region is where the antibody binds. So up here in the top, that's where antigen is going to bind to the antibody and stuff, and it's called the thermocomplementary determining regions, or CDRs. So this is where your antigen will attach to the antibody to alert the B cell that there's something there, and you need to produce more antibodies or to do what antibodies do to fight back in the body. You might wonder, if there's only kappa and there's only lambda, how can we have so many different antibodies for so many different diseases if we only have those 2 parts? And well, it's coded in the gene in multiple genetic segments. And the way in those segments are translated, they're put together in different ways. So it's kind of like alternative splicing, where if you have a gene A, gene B, gene C, you can have A, B, C to have one, or C, B, A, or B, A, C, or any of those combinations out there to form different antibody variable regions for different antigens for all that you need for your immune system. So that's the light chains. So going on to the heavy chains. The heavy chains are the big, long ones of the Y. These right here. They're also identical chains. They're exactly the same. They're divided into 4 parts. We have the variable region up on the top that's involved in binding. And then we have constant 1, 2, and 3 on both sides. There's 5 isotypes. Each one determines the class of the antibody. So mu is for IgM, delta is for IgD, gamma is for IgG, epsilon is for IgE, and alpha is for IgA. So whatever you have coded here for whatever, if it's mu, it will be IgM. That will be the antibody. So it determines the class of whatever it is. And also on this part, to talk about this here, is we have what's called the hinge region, excuse me. And that's what causes the Y part of the antibody to be formed. And in between holding your heavy chains and your light chains, you have disulfide bonds, and that's what holds them all together and forming the overall antibody shape. And so here's my sources. So I use Science Direct, mostly for the immunoglobulin domains, the textbook, Cubo Immunology, and Dr. Blair's videos and lectures. So thank you for watching my video, and I hope you enjoyed it.